Okay. Just a minute. We'll start in a minute. Okay. Hello. Hi. Okay. So now we are good to start. I hope last weekend things are now clear. The trial feasibility we discussed, right? The clinical trial planning and timeline on the Saturday. And on the Sunday, we discuss about the trial feasibility. I hope both of the things are making sense to you. The next topic is the study design and methodology. So today we are going to discuss about the study design and methodology. Let me share screen with you so that you can be able to see the title. I hope you have seen in the link which is shared because we mention the topic of the session in the links only. Just a minute. I hope it is visible. May I know? Yes or no? Okay, thank you. If any glitch in between, if you found, please, if you are finding any glitch in between, please feel free to drop messages in the group, okay, so that we can solve it. Because it is an online session, we need to take care a little bit. I hope the recording is on. Yeah. Okay, see the title. So now this is the complete new, module or new part you can consider for the whole journey of clinical, research and project management, clinical trials and project management. But this is very important. Once you have the timeline idea in your mind, when you have feasibility assessment in your mind, now you have idea about protocol and all the things are now clear. Now, the next question which comes into the pathway, that is the design, okay? The study design, the methodologies, and the clinical research. So, today we are going to discuss one of the most important topics you can consider in the clinical research, and which is the study design methodologies, okay? Before conducting any clinical research, you need to understand this. The first and the most critical step is deciding how the study will be conducted. What will be the method, right? We have lots of methods in this particular field. So this planning process is known as the study design. Okay, the first and most critical step you can consider of deciding how to, how the study will be conducted, what will be the methodologies for it, and this planning, is known as the study design. So think of the study design like the blueprint of some very important work. Okay, so it is the whole methodology. Okay, similarly, you just need to understand that study design is, if the study design is poor, study design is not appropriate, the results of the research may be unreliable. Okay, it may be biased as well, or even completely wrong as well. So, as a future pharmacist, as a future family professionals, clinical research coordinators, medical writers, pharmacovigilance professionals, I don't know what name I should give to you, but yeah, the meaning is simple, that you are going to work at anywhere, right? Or the simple clinical data managers, you will frequently read and interpret these clinical studies, and that's the reason understanding study design is essential for us. And that's the reason behind this title. This is the reason behind why we need to discuss this topic today. Okay, your why should be very strong. Okay, you need to have one very strong reason with you to study this, the whole topic. So today's session will help you to understand what. Why, first of all, why study design matters. What are the different types of the study designs we have? Okay, the broadly it is classified as the experimental studies and observational studies. Also, experimental is known as the interventional studies. I will let you know. So broadly, it is classified as observational and experimental. Also, we'll understand about the clinical applications of the each of the studies, how the researcher choose the best design for a research question. That is very important. Question that, yeah, I know the types of the study, but how can I decide that what is the best purpose for my study, right? So the lastly we'll discuss that what are the clues to identify the best design for the research. So at the end of the whole session, whenever you read a journal article or something which is published, you should be able to identify what type of study was conducted in this particular uh, you know article if you are reading or study you are observing, and understand its strengths and limitation, both of the things. Okay, so that is the overall thing. Okay, this is the title, this is the meaning of the title. Is it clear or not? So I can move forward with it, but first you need to understand the title itself. If I will get some thumbs ups, I will move forward, otherwise I will explain it again. Okay, so yeah, it is making sense, right? Let's discuss about learning objectives. You know now what does learning objectives means to us. Learning objectives means by the end of the session, what should I expect? Okay, what I will be getting at the end of the session. So by the end of this whole session, you should be able to first left side, downside, you can see define the study design. You should clearly understand what type of study designs are there in the clinical research, what it means, and why it is necessary in the clinical research. Second objective is by the end of the session, you should be able to differentiate some major study designs. You should be able to differentiate between what is called experimental, what is called observational, and identify some subtypes as well. What is randomized control trial, what is cohort studies, what are case control studies, what are cross sectional studies. All the things will be clear to you in the second learning objectivities. The third learning objective is to be able to understand some of the advantages and the limitations of the each of the study. No study design is directly perfect, you can say. Each has its own strengths, each has its own weaknesses and appropriate applications as well. So you should now know that which of the when each the design is useful, will in which scenario what type of the study will be helpful to us, right? Based on that we can select. The fourth learning objective is by the end of the session, you should be able to select some appropriate design, because to know is not our final goal. Okay, that is, it is very easy to understand the definition and all, strength and weaknesses. Once you will encounter, you will understand that it is very easy, right, to theoretically able to understand. But the most important learning objective is the fourth, selecting appropriate design. When I say this, let me tell you, many of our students are not able to select appropriate designs for their particular project. That is what my teaching objective behind putting this module in front of you. I want your clinical clarity about selecting appropriate design. I don't want that you will please memorize the definition and all. Nobody is going to ask you define what is cross sectional, define what is cohort, unless and until you are in a viva, right? But in real life setting, what happens, people will come to you and ask you that, see the proposal, see this protocol, see this rough protocol, raw protocol, which is not finalized yet, and do let me know which type of study will fits into it. Okay, that is a practical application. Okay, so researchers choose study design based on their objectives, research objectives, ethics, cost, time, disease prevalence, all the things. I will let you know how to identify it. That is our fourth learning objectives. And you should understand this decision making process, right? And the last learning objective is to be able to interpret research literature. So, as a healthcare professionals, you will read some clinical trial and scientific publications. Understanding study design helps you to further, critically evaluate the evidences before applying it to the patient care. So that is what our learning objectives are. Are the learning objectives clear? May I know? Okay, so let's start with the very first learning objective. What do you mean by the study design? Okay, let's understand the basic definition. Definition is for only our understanding, guys. Okay, do not try to memorize anything. Go on a concept clarity. Okay, even if you are not able to define what the study design means, that's completely okay. You should know that what are the, what does exactly it means. Okay, so basically study design means a structured plan, because which is standardized, pre-standardized, pre-decided, right? Because nobody can stand and tell that I want to do this study this way. It is not appropriate. Okay, we need some of the standard approach. We need some structured plan. Okay, so the definition it itself, it is a structured plan used to answer some of the big research questions, I can say. So in simple terms, you can just have idea. Study design tells researchers who will going to participate in the study, what intervention will be given, what outcomes will be measured, how long participants will be followed. All those questions will be answered in the study design. Okay, if I will choose experimental, the whole questions answers will be different, right? Answer of the question will be different. If I will choose observational and the subtypes of it, the again the answers of these questions will be different. So the useful framework, that is very important, which is known as a PICO. Okay, PICO stands for population, intervention, there's a comparator, and the outcome. Timeline is also added later, but you can just remember by PICOT. So who, population means who are studying. For an example, adults with type 2 diabetes mellitus, it is your population. Their study design will define that who will be participating, who are we studying. Okay, intervention, what treatment is being tested, that is also mentioned in this study design. What treatment is being tested, that is intervention. For an example, some new anti-diabetic drug, right? So that is the in intervention. Then we have comparator, what are we comparing against. Okay, for an example, are we comparing some placebo, or are we comparing some standard therapy which is already available and we are comparing with, for an example, metformin. Metformin as an anti-diabetic drug is already available and we are synthesizing some new molecules and we are comparing with them. So that is comparator. Okay, so that is also a study design question. O is outcome, what are we measuring. For an example, if the study is related to the anti-diabetic, we need to have an outcome, that is reduction in HbA1c, right? And the T is timeline, for how long the study will be conducted. For an example, 24 weeks, right? So that is what the study design means. Okay, so the clinical example also I can give to you, but I will let you know in the upcoming slides the examples and everything. For now, just understand study design is not something which can define in a one single line, and it is not something that is small approach. Okay, it covers the whole background. It is setting the base for any type of clinical research. That is what the study design means. It is a structured plan to answer some research question. Answers, okay, who are we studying, population, what treatment is being tested, intervention, what are we comparing against, comparators, what are we measuring, outcomes, and for how long, that is the timelines. Everything is covered here in the study design. Now let me tell you why this study design is so important to us, why we are studying this. Let me tell you guys, many students underestimate the importance of study design. You need to understand, even the most expensive study can fail if the design is poor. Of course, if you don't know what to compare, if you don't know how long the study can go, if you don't know that who will be your population, appropriately, study can fail, right? So let's discuss the reason. First of all, validity. What do you mean by validity? Are we measuring what we intend to measure? That is what the validity means. Are we measuring what we intend to measure? So, for an example, understand by the example only. If the hypertension, study that aims to evaluate some blood pressure reduction we are performing, and the design should accurately measure the blood pressure changes, right? Whatever chemical moiety you are having for the clinical research, that should accurately measure what? Blood pressure changes. Poor design may lead to some incorrect conclusion in the whole scenario, guys, right? That is what the validity. We need to focus on blood. This is a very simple example I'm putting in front of you, not the complex one, but you can imagine the scenario with the validity to the complex one. Okay, so in that it will be going very wrong, right? Then second thing we have, that is a reliability. Reliability means consistency, right? So that is a very important term. If the study is repeated under some similar conditions, the result should be also similar. right? Reliable studies generate some trustworthy evidences for us. So that is also the reason why study design definition and study design is so important to us. Then we have reproducibility. Reproducibility means other researchers should be able to repeat the study and obtain some similar findings in the future as well. Okay, this is the cornerstone you can consider for scientific research, guys. I will let you know everything in the upcoming slides with examples. This is only the basic thing for our understanding why the study design is so important, right? If there is no study design, you want having some validity, reliability, reproducibility, right? And then regulatory acceptance. Okay, we have regulatory agencies such as FDA, EMA, CDSCO. We discussed about this in the previous week, right? So it requires some, very long and robust study designs before approving some new medicines. And we required study design in a very accurate form as well. Okay, so just imagine a scenario where the company is claiming that its new antibiotic is highly effective. If the study design was, poor, doctors may prescribe an ineffective medicines to thousands of the patient, right? That is also the reason why study design is matters at the patient safety level as well. So this definitely could lead to some resistance, failure, patient harm and all. That is the reason study design directly impacts the patient safety as well. And that's the reason to study, right? I hope now you have idea what is study design, why we are studying, what are the objectives to study. Okay, the base is clear. We are ready to move forward with the classification of the designs. So let's come to understand the broad classification of study design. All the clinical research studies can generally be divided into two major categories, whether it can be experimental, interventional also you can consider, and the another is observational. Very simple thing to understand. In the observation, I am only going to observe what is happening in the patient scenario. In the experimental and interventional, I am going to do something into the therapy, maybe the change, maybe the reduction, maybe omission, maybe addition, whatever it is. That is a simple meaning as the name of the studies. Okay, in the experimental, the researchers actively intervenes into it. The investigator will be further deciding which treatment participants will receive, how the intervention is administered. For an example, a clinical trial which is evaluating some new cancer drug, for an example. So the researchers will assign the participants to receive drug A, drug B, placebo, whatever they are comparing. And these are called interventional studies. The intervention is there. The exact opposite term is observational studies. Researchers do not assign treatments in the particular observational study. Instead, they only observe what naturally happens, right? For an example, you are the researcher and you are studying some smokers and non-smokers and observing that who will develop lung cancer in the next upcoming years. You are not doing something inside it, right? You just observe. So no intervention is given. Researchers only collect and analyze the data. Okay, so that is how you can simply remember. Okay, so observation, you simply observe. In the experimental and interventional, you need here the intervention, you need to do some interventions into it. Okay, so experimental studies, you can directly imagine that it is stronger evidence for the causality, because of course you are going to intervene something, it will be more expensive as well and more complex as well. While on the other hand, observational studies, easier to conduct, less expensive, and it is useful when the randomization is unethical or impossible. You will get it the last sentence I told you, which is in the upcoming slides when we are going to discuss about RCTs, randomized control trials. Okay, but don't worry, observational study and experimental study, I hope it is clear to you that what is the definition of it. Are you able to understand this? Yes or no? If any questions are there, you can put your questions into the chat box. I'm not providing you the access right now to unmute yourself, guys, because we have 15 people. So I don't want any unnecessary disturbances in between. But you can write your questions into the chat box. We will address at the end of the session as well, and then the middle of the session as well. Okay. So I hope the definitions are clear. Let's start with the experimental study design and understand it. Beginning, let's begin this whole experimental study. design concept with the first major, category. As a first major category you can imagine, and that is the experimental. First we will understand the complex one, and then we will understand the observational, which is simple. Experimental studies are considered, you know I can say, most powerful, guys, most powerful approach for evaluating any type of, effectiveness of the drug and safety of some medical interventions and all. For all that, you need to do some experimental study, which is also known as interventional studies. The defining feature of this particular term is that the researchers actively assign a treatment or intervention. Researcher assigns the treatment or interventions, unlike observational studies, where researchers are only going to observe the things. Here, the investigator controls what participants will receive, while in observation, there will be no control over, it. You will be only seeing it, okay, and observing it. Now, in the experimental, three key characteristics are there. Very first is, here the researchers are going to assign the treatment. So the investigator will be deciding which participants receive new drug, which participants will receive standard treatment, which participants will receive placebo. So, this control allows the researchers to compare the outcomes also objectively, right? So that is understandable. Second thing, it is in a controlled condition, guys. Obviously, everything is pre-planned here and everything is standardized here in the experimental and interventional. We cannot do without planning, and we cannot do without standardization the experimental studies, right? So eligibility criteria, treatment duration, dosage, follow up schedule, outcome assessment, everything, like, everything will be planned and standardized when you are doing an interventional study. So that, further reduces some unwanted variation into the study. That is the reason why control studies are important. And then measures outcomes. Researchers evaluate whether the intervention produces the desired effect or not, whether it is, satisfying our goal or not, right? For an example, we want the reduction in the HbA1c, whether it is happening or not, reduction in blood pressure, whether it is happening or not, right? So for an example, if they are studying some anti-cancer, tumor shrinkage will be your measure outcomes, improved, survival will be your measuring outcomes. So this is how it goes. Researchers assign treatment and intervention. It will be on a control condition, and they are going to measure some outcomes. Okay, so this is the overall experimental study. For an example, understand, suppose a pharmaceutical company develops some new antihypertensive drug. Researcher will recruit 500 patient. Group A, he or she will decide. Group A receives the new drug. Group B receives the standard treatment, whatever available antihypertensive. After 6 months, blood pressure reduction will be compared. Because the researchers assigned the treatment, this is called an experimental study. I hope this example is making sense to you. It is easy for understanding, right? Are you able to understand this? Okay. So, why, let me just give you one more idea here only. Why are these experimental studies so important for us? Because they provide some strongest evidences for the establishing. Just imagine the antihypertensive wala example. Okay, you can directly relate what was the cause and what is the fact, right? So, for an example, instead of saying that people taking drug, something like drug X seems healthier, we can say why this experimental study, drug X causes the improvement. This is why regulatory agencies, prefer the evidences from experimental studies when it comes to the drug, guys. Okay, so of course, most of the drug approvals worldwide are based on the experimental clinical trials only. When evaluating some new medicines, pharmacists must understand how the study was designed, whether the findings are trustworthy or not, and whether the results can be applied to the patients or not. But just for your information, remember, most of the drug approvals worldwide are based on experimental clinical trials. So what comes into experimental trial, guys? What type of studies are there in the experimental or interventional type of the studies? Very first is randomized control trials. I told you three approaches. Researcher is going to intervene into it. It will be on a controlled manner, right? And the measuring outcomes. Very first is, on the basis of that scenario only, like the three major pillars only, they have designed a very first experimental interventional type, subtype you can consider, and that is randomized control trial. You can see the middle word and the last word, control trial. right? But randomized. Okay, the most important experimental study design, most important interventional study design is RCT, randomized controlled trial. And this is often called as a gold standard of clinical research, guys. Okay, I have asked this question many times in the interviews and the vivas and all. What type of the study is called as a gold standard of clinical research? You have now idea that in the most of the clinical research, when it comes to the drug, right, always the experimental and interventional studies are preferred, right? And we have subtypes into it in the experimental and interventionals. And the very first is RCT, and that is the gold standard of clinical research. What is an randomized control trial, guys? Okay, focus on the word. It is randomized. Participants are randomly assigned to different treatment groups. Randomly. The word randomized means assignment occurs, by chance here rather than the investigator's choice. Okay, rather than the investigator choice, by chance. For an example, I will give you the examples of orthopedic device here. For an example, if you know, nowadays some silicon gadgets are, coming in the market for the for replacement of plaster of Paris, because POP, plaster of Paris wala jo hota hai, that is restricting movements and all, right? So they are coming up with some silicon gadgets so that the movement restriction can be reduced, and it is very much convenient as well rather than that plaster of Paris wala things, right? So for an example, if the RCT is happening for some silicon devices in orthopedic ward, randomly patient will be assigning to the plaster of Paris and silicone, devices, and then collectively measuring the outcomes at the end of the study period. That is what is known as randomization. Randomization can be, again, इसमें भी subtype आएगा, randomization can be also blinded, double blinded, triple blinded and all. I will let you know what does it mean, but for now understand, participants are randomly assigned to different treatment group. Okay, and this is similar to, like, for an example, tossing a coin. Okay, tossing a coin, participants have equal probability of entering either of the groups. Okay, typical structure of this particular RCT trial is, for example, some group A will receive the new drug from, for an example, group B will receive some placebo or some standard therapy. Researcher then compares the outcome. That is what the randomization means. So why randomization matters, guys? What is the meaning of doing this? Without randomization, guys, one group might unintentionally, contain some very much younger patients, or maybe very much healthier patients, maybe, or maybe patients with some very few comorbidities, right? So these differences can influence the result, and we need unbiased result, so that we are choosing randomization, which is going to be balance these factors. Okay, that is why the name is randomization, to avoid biasness. Yes, you write it, the correct thing, right? So these differences can influence, right, the results, and we don't want it. For an example, suppose we recruit 200 diabetic patient, randomly assignment, creates 100 patient to new drug and 100 patient to standard treatment. Because the assignment is random, baseline characteristics are usually similar, right? And this improves study validity. It is not like 100 we are deciding, but which patient enters into this 100 category, we didn't decide, right? So once the 100 patient will receive the group A, the new drug, and the 100 patient receives the standard treatment, the study will close. So characteristics will be usually similar, and this improves the study validity. That's the reason why randomization will be important. Why this called as a gold standard? That is important question. Because it is minimizing the selection bias, guys. It is co-founding variables, and it is, investigator influence also, it is minimizing, right? Investigators are influenced, there is nothing, because it is all randomization. Investigator cannot decide who will take which drug. That's the reason randomized control trials provide the strongest evidences regarding the drug efficacy. Okay, and we have some of the very real life example with us. We have Pfizer and BioNTech COVID-19 vaccine trial. That was a large randomized control trial, involving thousands of patients. The study was demonstrated vaccine efficacy before the regulatory approval, guys. Okay, so whenever a new medicine enters into the market, pharmacists should ask always, was the efficacy demonstrated through the properly conducted RCT? If not, evidence may be weaker, very weak in this particular scenario. So that is why RCT matters. Let's discuss the components of RCT. Every randomized control trial contains some several, essential components, guys. For example, randomization. Participants are assigned to groups by chance here. Okay, not by choice. Purpose is through selection, reducing selection bias and balancing known and unknown co-founders. For an example, some random computer generated allocation will be done. Second is a control group. A control group, serves as a comparison, right? Possible control includes some placebo control, inactive treatment, sugar pills, right? And also it contains some active control like some standard treatment, new antibiotic versus, for an example, amoxicillin. So that is control group. Okay, why control groups are needed? I hope you have idea, because without comparison it becomes difficult, right, to determine whether the improvement occurs because of the drug effect or some, it is some natural recovery. We need to understand with the control group, right? And then blinding means, I can say, blinding means concealing the treatment allocation. Okay, we need to hide. The purpose is to prevent the bias from the patient, from the investigator, from the outcome assessor. Nobody can interfere. That is why blinding is important. We will discuss blinding in detail in the next slide. Okay, and then we have prospective follow up. Prospective means researchers move forward in time. Okay, prospective, that means researchers are going to move forward in time. Patients are followed, after the treatment assignments, and researchers will observe the outcomes as they occur further, right? For example, enrollment is done, randomization is done, treatment is done, and then follow up and then outcome assessment. So this forward looking approach improves the data quality. That is what the prospective follow up means. Okay, for example, if the cholesterol lowering drug का कुछ trial हो रहा है, for example, cholesterol lowering drug trial is happening, recruiting the patient is the first step, randomizing participants, then, the second step, administering the new treatment, follow up for 1 year, and then comparing their LDL reduction report, low density lipoprotein report. So that demonstrates the all four components altogether: randomization, control group, blinding, and prospective follow up. But always remember, an RCT is not simply a study, involving a treatment, guys. It must include randomization, it must include control group and prospective follow up. These features differentiate randomized control trials from many other designs. Okay, I hope you will remember this. Okay, in RCT also we have some types. Okay, so in RCT also we have some types. So I hope you are making notes. We have broadly classified experimental and observational. Now we are discussing experimental, which is also known as interventional, and we discuss further inside it the very first randomized control trial, and now the types of randomized control trial. You can make some graph or something if you want, or that what do you say, the arrow karke you can just understand the things. Okay, someone is messaging me that due to poor network and rain, I am unable to join this. Okay, that's fine. No worries. So that is what we are discussing. Types of the randomization. Let's discuss how randomization actually be performed. Different methods are used depending on the study objective and sample size. Depending on is very important word, guys. Depending on which is your objective, and what sample size you are targeting. So very first we have is a simple type of randomization. Left side, down side, you can see simple randomization. The simplest method, as the name suggests, for an example, a coin toss. Okay, coin toss, या फिर for an example some random number table, या you can say some computer generated allocation. Each participant has some equal probability of entering any group. Okay, so patient 1 receive the drug, patient 2 will receive placebo, patient 3 will receive drug, patient 4 will receive again the drug, patient 5 will receive the placebo, right? So assignment occurs completely by chance. Who will be entering at a patient 1, 2, 3, 4 number, that will be completely different, right? So the only limitation of this simple randomization is assignments, occurs completely by chance here, but groups may become unequal in the smaller studies, guys. Okay, if your sample size is less, some inequality can happen. There is some risk inside it. That is what the simple means. To avoid this, we need a second type of randomization, which is a block randomization. that is used to maintain some equal numbers in each group. That was the side effect for simple randomization. Block means it is used to maintain some equal numbers in each of the group. For example, block size 4. That means possible sequence will be A, or A, B, right? Or B, A. So that ensures some balance throughout the enrollment. Okay. So no inequality will be, you will be facing in the block randomization. Okay. Block size 4, block size 6, block size 8. In the bigger studies, you will be getting some big blocks as well. So frequently used in the multi-center clinical trials, the block randomization. The third is a stratified randomization. Participants are first divided into some important subgroups here. Okay, participants are first divided into some important subgroups. Stratified karna. That means dividing them into some important subgroups. For an example, male, female, or diabetic, non-diabetic, younger, older. So then the randomization occurs within the each subgroup. Okay, more equality will be identified. Now why this is stratifying is important, because to ensure balanced distribution of critical characteristics like diabetes and all. Okay, so it is all more important as compared to the block and the simple one. Okay, for an example, if the diabetes influences the outcome, researchers ensure the equal diabetes representation in both of the treatment groups, right? That is what the stratified means. And the last one we have, that is a cluster randomization. Entire group are randomized instead of the individuals. For an example, this hospital, or for an example, this village, for an example, this school. So this village receives the placebo. This village, village B will receive the standard treatment. So that is a cluster, big randomization. Entire groups are randomized instead of the individuals. For an example, hospital A received some new infection control protocol. Hospital will B receive some standard protocol. This is useful when individual randomization is impractical. Okay, so understand, simple means individual random assignment, nothing is there. Block is balanced numbers. Stratified means balanced characteristics, and cluster means groups randomized. Are you able to understand this? In a very simple and lame language, I am explaining to you whether you are able to understand this. So if you want to do interventional studies, if you want to do RCT Institute, randomized control trial, based on your study objective, what do you want to assess, right? Based on that and based on your participants, you can identify which type of RCT do you want into your study. Are you able to understand this? Yes or no? Only one thumbs up? Two? I have 15 people with me. Three, four, five. Okay. Okay. So this is very important, guys, because nobody is having idea that based on what the study design is being identified, and it is being fit into the title. Okay. Now let's understand what is blinding in the clinical research, because blinding can happen at any level, right? So blinding is one of the most important concept. Blinding means, you can say, keeping the treatment allocation hidden. Okay, now किससे hide करना है? Okay, if you want to hide from one person, it will be the term for it. If you want to hide from three persons which is involved in the study, the term is different. Okay, so blinding means keeping treatment allocation hidden. The purpose is, we don't need a bias here. Bias means some systematic error that affects the study results, right? Very first we have, that is a open label. Open label means it is open, everyone knows the treatment, participants will know, investigator will knows, right? For an example, comparing some survey results versus some medications. Okay, so blinding may not be possible in the survey versus medicines. Open label it should be. Okay, what do you mean by single blind? Single blind means participants does not know the treatment assignment, investigator knows. Okay, only participants are not aware. That is a single blind study. For an example, patients receives either placebo or active drug, but does not know which is the thing they are receiving, whether the placebo or whether the drug. But investigator knows. That is single blind. What do you mean by double blind? Neither the participants nor the investigator knows what is the treatment is being assigned to the patient. This is the most commonly preferred design, guys. Double blind study. Benefit is reducing patient bias, reducing investigator bias as well. For an example, pain medication trial ho raha hai. Okay, for an example, some pain medication trial is happening. If the patient know they are receiving, the new drug, they may report some, greater improvement by chance, psychological influence. If investigator know the treatment allocation, assessment may be unintentionally also influenced by the psychological things. That's why double blinding prevents this type of things, right? Neither the patient nor the investigator is aware that who is receiving what type of treatment. Triple blind is also there. Three parties remains unaware. Participants, investigators, and third, data analyst or the outcome assessor is also not aware कि इस patient ने कौन सा treatment ली है. So that provides even greater protection against the bias. Okay, so open label, everybody knows. Single blind, participant doesn't know. Double blind, participant, investigator not knowing. Triple blind, participants, investigator, data analyst also combinedly not knowing what is happening. For an example, COVID-19 vaccine trial. So that is often uses some double blind methodology, guys. Participants did not know whether they received vaccine or whether they received placebo. Investigators also remain unaware. Why blinding is important? I hope it is clear to you. Like, without blinding, patient may expect improvements. Investigator also may, we can say consciously or unconsciously influence the assessment. And this can be distort the results, and we don't want that. Okay, so when you are reading some published literature, always check that was the study be blinded, was it double blinded, was it open label. Okay, this information helps to determine the reliability of the study finding. Okay, I hope it is clear to you. What do you mean by parallel design? This is also one of the big concept to understand. So let us discuss one of the most commonly used clinical trial design known as parallel design. So in the parallel design, participants are randomized. It is also RCT. So it is, participants are randomized into some different groups and then remain in their assigned groups throughout the entire study. That is parallel design. Again, I am repeating what is happening here. Participants are randomized into different groups and remain in their assigned group throughout the entire study. They do not switch the treatments. How it works? For an example, if the group A receives the drug A, followed until the study completion, and if the group B receives some placebo, they are also following until the study completion. Both groups are observed simultaneously and the outcomes are compared at the end. So that is known as parallel because both treatment groups run side by side or in the parallel. That's why they have named as a parallel study. Okay, so advantages are it is very simple design, easy to plan and execute, easy data analysis is there, suitable for the most of the diseases like hypertension, diabetes, cancer and all, and no carry out effect is required here. Since the patients never switch the treatments, previous treatment cannot influence the future outcomes inside this parallel study. The only limitation with this parallel study is it requires some larger sample size, guys. Each participant contributes to the data for only one treatment. So it is more expensive, right, as compared to other studies. And more participants means the higher cost, right? So you need to understand. For an example, if we compare to the COVID-19 vaccine phase 3 trial, that, most of the COVID-19 vaccine phase 3 trial use a parallel group design. Participants receives either vaccine, either placebo, and remain in that group throughout the study. Okay, so when you are reading some drug efficacy trial, the majority of the studies you encounter will use some parallel design because it is very practical, reliable, and regulatory friendly. Okay, so I hope it is making sense to you that what do you mean by this parallel design. So this is all about randomized control trials and blinding and parallel design concept. The next thing we have in the experimental and interventional, guys, and that is a crossover design. So let's move to some another important design called crossover. So unlike these parallel studies, parallel simultaneously goes and participants were remaining into the particular assigned group till the end of the study. It is completely the opposite term. Okay, participants receives more than one treatment during the study, more than one treatment during the study. Essentially, each participant act as own control. Okay, how it works? For example, in the phase 1, group A receives drug A and group B receives the drug B. But they will be assigning with some washout period. Okay. And then it will be switched to group A receives group, the drug B, and group B receives the drug A. What is washout period? Washout period is a break between the treatment, guys. So purpose is very simple, to allow the first drug to leave the body completely before the second treatment begins. Without washout period, effects of the first treatment may continue, right? And that affects the results as well. So that is called carryover effect. Okay. And we don't want carryover effect, so we are following a washout period. Okay. So for an example, understand crossover design with an example. For an example, suppose researchers are comparing drug A for migraine and drug B also for migraine. Okay, two drugs. Patients receive drug A for 4 weeks. Okay, then a 2 week washout period will be provided to them, and then the patient receives drug B for another 4 weeks. And the same patient experiences the both of the treatment. That is known as a crossover design. Advantages, you need here a smaller, very smaller sample size. Each patient serves as their own control. So this reduces variability, right? It is also cost effective because fewer participants are there. So cost will be saved, and it increase some statistical power as well. Differences between treatments are, easier to detect. The only limitation of this crossover design is, it carries some time after washout period, carryover effect, residuals, drug effects that may influence the outcome. Longer study duration, because washout period dena padega, participants undergoes into multiple treatment periods, and it is not suitable for all of the diseases, right? It cannot be used when the disease is very progressive or the condition changes permanently and all, right? In the small cases, you can do some crossover designs. Okay. Like for an example, cancer, a patient cannot return to the baseline after the cancer progression, right? So we cannot use some crossover design in that particular situation. So what is the clinical application of crossover designs? For an example, some small type of pain management studies, asthma studies, migraine trials, pharmacokinetic studies, that's where crossover designs are useful. Crossover studies are frequently used when you are, comparing some different formulation, comparing some generic versus branded products, bioequivalence studies. So this is particularly important for the pharmacist working in some regulatory affairs and clinical research, right? So that is very important to understand crossover. Now the next design we have in the experimental and interventional is a factorial design. What do you mean by it? So let's discuss factorial, one of the most efficient study design in the clinical trial. Okay, factorial. Name से ही पता चलता है, the name itself has the meaning. A factorial design evaluates multiple interventions simultaneously within a single study. Okay, factorial, multiple factors, multiple interventions simultaneously within a single study. Okay, so instead of studying one intervention at a time, researchers can investigate two or more interventions together. So that known as a factorial design. Test multiple interventions simultaneously. Example से समझते हैं. For an example, suppose researchers wants to study some vitamin D and calcium. Okay, both. Participants are randomized into four groups: group 1, 2, 3, and 4. Group 1 will receive vitamin D plus calcium. Okay. Group 2 will receive some vitamin D only. Group 3 receives only the calcium, and group 4 will receive neither treatment, like placebo. Okay, so what can researchers learn by this factorial design? Researchers can determine effect of alone vitamin D, effect of alone calcium, combine effect, of both, all within a single trial. That is known as a factorial design. Okay, any design can be factorial, guys. Okay, it is not a separate, right? So you can just understand any RCT also can be factorial design. Okay, highly efficient it is because multiple questions answered simultaneously into factorial design. It is cost saving because one trial instead of multiple trials are happening into factorial design. Fewer participants because you are comparing with the, separate studies only in the particularly defined population or participants. For example, understand cardiovascular prevention trial. Cardiovascular prevention trial. Researchers may evaluate the aspirin effect and some cholesterol lowering drug effect as well. Statin ले लेते हैं, for an example, atorvastatin. At the same time, if they are doing, so this allows investigation of aspirin alone effect, statin effect, and combined therapy effect. Okay, so that is known as a factorial design. Limitations are only, little bit on a large extent, it is little bit complex. Planning and analysis are more difficult. Interaction ho sakta hai, right? So treatment may interact with each other. Like for an example, one intervention may enhance or reduce the effect of another one. That is the risk factor. So factorial designs are increasingly used in what type of studies? Like for an example, some prevention studies, nutritional research because you want some synergistic effect, or some cardiovascular research or public health intervention. Right? Pharmacists often encounter a combination therapies, and for that, factorial word needs to be there in your mind. Okay, that helps to evaluate whether the combinations provide benefits beyond the individual drugs or not. Right? So I hope it is making sense. Now, let me tell you some clinical trial designs across the drug development. Let's connect study designs to the drug development processes. So, before the medicine reaches to the patient, guys, it passes through some several phases of clinical trial, and you know that each phase has different objective, and it may use some different study design as well. I told you that we discussed the phases, right? In the, I think in the very first session of mine, I discussed with the this phases. Like, very first is the primary objective is to assess the safety, right? So questions will be answered: is the drug safe? What does what will be the dose of the drug should be used? How is the drug absorbed? What adverse effect will be occurred? Safety, right? So first in human trial of the new anti-cancer drug, for an example, that is phase 1, right? So of course, based on that, you will be deciding the study design. Okay, if you want phase 2, that talks about efficacy and continuous safety, you will be answering: does the drug work, or which dose is best? So common designs will be included: what? Here, some randomized control trial will be needed in phase 2. Some parallel group studies will be needed in phase 2. I hope when I say randomized control trial and parallel group, you are, it all making sense. In a phase 1, dose escalation study and open label studies only will help. Okay. And in phase 2, some RCT trial will be required. Okay. Then phase 3 is, confirming efficacy and monitoring safety. What will be the common designs? Like some large RCT will be required, double blind trial will be required, multi center studies will be required. Okay, and when it comes to phase 4, then phase 4, the purpose is to detect some rare adverse effect, post marketing surveillance, right? Real world effectiveness and all. So here the whole thing will be little different, right? So this pharmacovigilance, I will let you know what to do next. But in phase 1, 2, 3, I hope it is making sense to you that what type of experimental studies will be required in different phases. Is this experimental study clear? The types, the names, the basic definitions? Yes, I can give you the examples of each of the phase, don't worry. I hope the definitions are clear to you. See, for an example, in phase 1, first in human trial of some new anti-cancer, I told you, right? So some dose escalation studies or open label studies will be required. For an example, in the phase 2, where you are assessing efficacy and, continuing the safety, uh, in which you will be requiring participants usually 100 to 300, right? And you will be a question, answering the questions like, does the drug work, or which dose will be the best? So you will require some randomized control trial and parallel group studies. For an example, I can say evaluating some glucose reduction in the diabetes patient. Okay, if you want to see that in the diabetic patient, if you want to evaluate glucose reduction, you require some RCT and parallel group studies in the phase 2, right? For an example, in phase 3, where you are confirming the efficacy and monitoring safety, you require some common designs, study designs like larger scale RCT, double blind trial, multi center studies. The very big example with us is the COVID-19 vaccine efficacy trials. phase 3 में RCT था, डबल ब्लाइंड था, मल्टी सेंटर स्टडी था, है ना? And then in the phase 4, you are monitoring some cardiovascular safety, you can imagine, for some newly approved diabetes drug. So you require a post marketing surveillance conducted after some regulatory approval only. Okay, and you will be detecting some rare adverse effect, long term safety, real world effectiveness in a phase 4. Okay, many serious ADR are identified only during phase 4, if you know. Okay, I can say some example. For an example, thalidomide teratogenicity was the phase 4. and some rofexosib cardiovascular toxicity also example of the phase 4. Okay, so I hope these things are clear to you now. In cluster class, we don't consider, no, that is completely depend on objectives, inclusion and exclusion criteria of the study. Okay, in cluster also your inclusion exclusion will be there. Okay, ऐसे नहीं है कि पूरा हॉस्पिटल आ गया, right? Can you clarify between experimental and interventional? Actually, this is interchangeable, interchangeably used words. But yeah, I already explained this, right? Experimental and interventionals. Okay, so now everything is clear related to experimental. Yes or no? Yes? Okay, so now we are all set to go for observational type of the study. We will now move to the second major category of the study design, which can be observational. यहाँ पे हमें सिर्फ observe करना है. We are not going to do any type of intervention. Okay, remember the key difference. Experimental study was researchers assigning the treatment, monitoring outcome, controlled manner. Okay, while observational study, researchers does not assign the treatment. Instead, researchers observe and analyze naturally occurring events. Okay, definition is very simple. Observational study is a, I can say the research design in which investigator collect the data without manipulating exposure or treatment. Patients receive treatments as a part of the routine clinical care. Researchers simply observe the outcomes. Okay, family students, if they are in a fifth year, you have idea. Okay, we have thesis and all, and we are only having rights to do some observational studies. Okay, why? What is the use of observational study? Sometimes experimental studies are unethical, expensive, and impractical. In such situation, observational studies becomes valuable. For example, suppose researchers want to investigate whether the smoking causes lung cancer or not. So it would be unethical to randomize patient into smoking and non-smoking groups, right? So that's the reason researcher will observe the existing smokers and non-smokers, and this becomes an observational study. Okay, the advantages are simple. It can give you some real world data. It reflects some actual clinical practice. It is less expensive as compared to RCT, randomized control trial. It is easier for recruitment. Participants continue the normal therapy. So, recruitment will be very easy. You are not going to intervene anything, right? And it is used for some rare outcomes. It can investigate some uncommon diseases as well. Okay. What is the limitation of observational is high risk of bias. Okay. Because there is no randomization, bias can be happen. And some confounding variables are also influencing, like other factors may influence the results of observational. For example, if the smokers are there, but smokers may differ from non-smokers in diet, lifestyle, and alcohol use, right? So we cannot blindly focus on the one single parameter. So these factors may affect the outcomes later, right? But let me tell you guys, we have some types of the observational studies as well. Types of observational study. You can remember in observational, it start with the O. So you can remember, like in the observational, we have everything related to C. We have 3C. Okay, C for cat, C. We have cohort study, we have case control, and we have cross sectional. Crossover is completely different. It is the experimental and interventional, guys. Okay, here we have 3C: cohort, case control, cross sectional. So this designs, form the foundation of epidemiological and real world evidence research. I will let you know each of the types in detail. So let's start with the cohort. What do you mean by it? Cohort. So let's discuss one of the most important observational studies, most widely happening worldwide, cohort study. The word cohort means group of people. Okay, group of people who shared some common characteristics and are followed over the time. Okay, same characteristic and are followed over the time. In the cohort study, researchers begins with the participants based on their exposure status and then follow them to see whether they develop a particular outcome or not. Please understand the concept. Here what happens, researchers begins with the participants based on the exposure status, what they are exposing to, and then follow them to see whether they develop a particular outcome or not. Okay, so basic structure is like start with exposed group already, for an example smokers. Okay, and then unexposed group, for an example non-smokers, and then follow the both of the groups over the period of time and observe who develops the lung cancer later, right? So the key feature of cohort is cohort studies start with exposure and move forward toward the outcome. Think of exposure and then arrow goes to the outcome. This is the hallmark of the cohort study, guys. For an example, research question it is answering, does the smoking increase the risk of lung cancer? Right? Simple. Exposure and then outcome. Researchers recruit, for an example, 5,000 smoker, 5,000 non-smoker, follow them throughout the 10 years, and at the end, comparing the lung cancer incidence later. Okay, so it can actually measure the incidence. Incidence means the number of new cases occurring during the period, right? So randomized control trial and cohort studies can calculate the incidence directly, guys. If you want to do interventional, the gold standard is RCT. If you want to do observational, the gold standard is cohort, right? So multiple outcome can be studied here. For an example, smoking may lead to lung cancer, smoking may lead to COPD, smoking may lead to cardiovascular disease. All can be assessed simultaneously. And strong temporal relationship you can identify. Like researchers know that the exposure occurred because of the disease. So this strengthens the causal interference, I can say, right? What is the limitation is it is time consuming because you require a follow up, maybe years ka follow up you required, right? Expensive, large sample size are often needed in this particular case, so it goes to many costs will be higher. And loss of follow up, participants may withdraw or become some unreachable at some time, so this can affect the results. Okay, so just give me a minute, guys. We have another session at 7:30. I need to drop a message. Just a minute. Okay, so in the cohort, we have a very famous example of this framing and heart study. If you know, that is one of the most well-known cohort studies in the our record, right? So many modern cardiovascular guidelines are based on finding from this particular study. That was cohort, okay? And you need to remember it is a group of people. Cohort means group of people and exposure to outcome it is, okay? Another type of the this observational study we have is a okay, in the cohort only, there is a subtype of prospective and retrospective. What do you mean by it? Cohort studies can be conducted into two major ways. Either it can be prospective, either it can be retrospective. Understand this difference. That is very important in the clinical research, guys. What do you mean by the word prospective? Prospective cohort study means the study begins today and it moves into the future. Okay, what do you mean by prospective? Today, the study begins and it moves into the future. Researchers will identify the exposed and unexposed individuals now, who is doing smoking, who is not doing smoking, who is exposed to the the important the opioids or something like that, if you want to check some another things, and who are not, and then follow them forward in the future. That is prospective. Today, like they are recruiting some smoker and non-smoker, follow them for 10 years and assessing lung cancer occurrence in both of the groups, right? So prospective has their own advantage: high quality data, better control, stronger evidences. The only disadvantage of prospective is it is expensive, long duration, as I told you the example 10 years, right? So that is the only disadvantages, limitations of prospective. On the other hand, we have retrospective cohort study. What do you mean by retrospective? Researchers use retro. Retro means historical, right? Researchers use historical records. Like both exposure and outcome have already occurred. That is what the retrospective means. Investigators look backward here using some existing data. Okay. For example, hospital records from 2010 to 2020, identifying smokers and non-smokers, and then today determining who developed lung cancer in the 2026. Okay, the simple retrospective cohort study has advantage. It is faster. You can come to the conclusion in 5 years, right? Or maybe in a year only you are having the outcome in your hand, because you are comparing some individuals who has today the lung cancer and then exposure. The reverse, retrospective, backward, right? The only disadvantage is you can find it some missing data, poor documentation, and limited control over the variables, right? Simple timeline comparison it is. If it is prospective, it is in the future, like present, in the future, and exposure is the outcome. In the retrospective, it is all about depending on the past records, outcome already known. Okay, so I hope it is now making sense to you. What is prospective? What is retrospective? Cohort can be both of the type. Prospective means follow forward, retrospecting means look backward using the records. Okay, so that is all about cohort. Now, the next observational study we have, that is a case control studies. Let's discuss some another major, observational study. Another C, case control. So, unlike this cohort studies, case control begins with the disease rather than the exposure. Okay, हमने उसमें क्या देखा था कोहोर्ट में, वी लुक फॉर एक्सपोजर एंड देन वी लुक फॉरवर्ड द आउटकम. In the case control, we look for, disease rather than the exposure. For an example, I will give you the examples, don't worry. Basic concept is researchers first identify the cases, individuals who already have the disease, okay, and then controls, individuals who do not have the disease, and they look backward to determine the previous exposure. Okay, the structure is like disease present, cases, then disease absent, controls, and then look backward. What are their lifestyle and diet things were there? They are only observing, they are not doing any intervention. That's why it is observational, but it is a case control, looking backward. Okay, for example, is the smoking associated with lung cancer? If you are doing some cases like patient with the lung cancer, and you are checking some control patient do not having a lung cancer within the same age line, and then investigate their smoking history backward. Okay, so case control studies moves with outcome first, and then observing their exposure in the past lifestyle. That is what the case control, exact opposite of what we understood previously, right? So this is very ideal for the rare disease, guys. Rare disease may take some decades to develop. Case control studies are much more practical in the rare condition identification. If the today the patient develops Alzheimer's, you can check later what was their scenario 5 years 10 years ago. It is also usually cheaper than the cohort study, right? Limitation is only the recall bias. Participants may incorrectly remember the past exposures. Patient can lie also, right? Like for an example, patient with some rare cancer may remember some smoking history differently than the healthy controls, right? So that is only the limitation of case control. We have historical clinical example in case control, thalidomide birth defects. Researchers compared infants with birth defect, infants without birth defect, and they discovered maternal exposure to thalidomide during the pregnancy. This landmark case control study changed the drug safety regulation worldwide related to thalidomide. Okay, so that was a case control study, guys. I am giving examples for your better understanding. Are you able to understand? It is simple concepts. Okay, I hope it is making sense to you, and everything is clear. Okay, cohort, exposure, outcome, right? Case control, outcome leads to exposure. And the next one we have, it is a cross sectional study. The third major observational design is cross sectional. Cross sectional study provides, cross sectional means a snapshot of population at a single point in a time. Think of it like taking a photograph. Photograph captures a moment, right, but does not show that what happened before or after it. Similarly, cross-sectional studies collect the information only once. For an example, it has some key characteristics like exposure and outcomes are measured simultaneously, and researchers do not follow participants over the period of time, right? The question it is answering is what is the prevalence of hypertension among the, for an example, among the PharmD students, right? So researchers, what they do, they will do some survey of thousands or 10,000 PharmD students today and measures the blood pressure and lifestyle factors among them. And at the same time, this is a cross-sectional study and taking a snapshot, nothing to do with after it or before it and nothing to compare, only taking a photograph. So what can be measured here? The prevalence. Prevalence means the total number of existing cases at a given time. For example, 20% of survey students have the hypertension. This is prevalence, percentage. So advantage is very quick, data collected only once, very inexpensive, right? There will be no expense into it, no follow up needed, and it is useful for public health planning. It is providing information about disease burden, right? But what is limitation? Cannot determine what is the cause, what is the effect, right? Because exposure and outcomes are measured simultaneously. In the cross sectional, it is only a snapshot. Okay, for example, if obesity and hypertension are found together, we cannot be certain which can come first with the cross sectional. That is only the disadvantage of it. But yeah, if you want to do some healthy surveys, if you want to check some knowledge, attitude, and practical studies, cap studies, if you want to, check some disease prevalence studies, quality of life assessment, cross sectional will be best option. Okay, so let's compare all together, comparing observational studies all together. Let's compare it. This comparison is extremely important. Cohort studies start with exposure. Direction is exposure first and then outcome, right? If the patient is doing smoking today, 10 years later, checking the lung cancer possibilities. Best for the risk factor assessment, disease development, and incidence checking. Okay, you can remember the smoking example all the time while learning the observational studies. It all makes sense. Okay, when it comes to case control, it starts with the disease, direction is backward, outcome first, checking the exposure later. Thalidomide, you can remember, that is best for the rare disease condition identification. Okay, क्रॉस सेक्शनल इज अ पॉपुलेशन स्नैपशॉट सिंगल पॉइंट इन टाइम प्रिविलेंस स्टडीज के लिए इंपॉर्टेंट है एंड इट इज जस्ट चेक फॉर एन एग्जांपल checking a prevalence of hypertension, right? So easy memory trick I can show you. Cohort start with exposure, who gets the disease. Case control start with the disease, what exposure caused it. And cross sectional start with current population, how common it is right now. Okay, so I hope all making sense to you. Choosing the right study design. So we have already learned the major study designs. An important question is, how do researchers decide which study design to use? So there is no single design, that works for every research question, guys. The choice depends on several factors. Very first, research objective. What exactly do we want to know? Right? For an example, if we want to determine whether the new drug works for whether the new drug will work or not, so definitely we want to choose RCT. अगर new drug का trial करना है, definitely we need to do some experimental study, and in experimental also we need to choose randomized control trial if we want to identify the whether the drug will be working or not. If we want to identify disease risk factor, we will choose cohort study, right? If we want to determine prevalence, cross sectional study. So research objective. Second, ethical consideration. Sometimes the ideal design is not ethical. For example, can we randomly assign people to smoke cigarettes? Of course not. So RCT is impossible. Okay, so researchers must use what? Observational studies. We cannot go for interventionals. Third, budget. Clinical trials are expensive. Large RCT may cost millions of dollars, and researchers with limited resources may choose some observational studies, maybe cross sectional or retrospective, because they are less costly. Okay, time. Some questions require answers quickly. Outbreak investigation, if you want to do, researchers cannot wait 10 years for a cohort type of study. Yeah, right? So, case control study may provide answers much faster. Okay, not written here, but let me tell you, fifth factor is a disease frequency, guys. If you want to check some rare disease, you can go for case control studies. Those are ideal. If you want to do some studies on common disease, cohort study may be feasible. Okay, so question like, does a new antibiotic cure the pneumonia better than the standard therapy? The best design will be RCT. If the question is, what factor increase the risk factor of the lung cancer? Best study design is cohort. If the question is, what is the prevalence of hypertension among the healthcare workers? The best design will be cross sectional. So when reading a research paper, always ask yourself, why did the researchers choose this particular design for this study? Right? Why should be very important. So a good researcher select the design based on the research question, guys, not the personal preferences or not by the suggestions of anyone. Okay, so I hope you will remember this. Study design selection examples, I already told you: drug efficacy, RCT, rare disease control factor, case control, and all. Right, I already told you these things. So whenever differentiating this research project, selecting the correct study design is one of the first decision made in the protocol development. You need to understand this. Okay. Strengths of evidence, pyramid. Now, not all scientific evidences carries the same level of reliability, guys. In evidence-based medicine, evidences is arranged in the hierarchy, and that is known as evidence pyramid. Okay, EBM, that is EBM is evidence-based medicine. Okay, for example, meta-analysis, highest level of evidences, combining results from multiple studies and providing the most precise estimate of treatment, right? Systematic reviews. Structured review of available evidences, randomized control trial, I already explained, cohort, I already explained, case control, I already explained to you, right? So the important clinical message is higher in the pyramid generally means less bias, greater reliability, stronger confidence. But yeah, it is expensive, right? So we have some real world clinical example. I think these examples I already told you in the slides, that COVID-19 we told, right? We discuss smoking, lung cancer, we discuss thalidomide also we discuss. So what are the key takeaway message, guys? Key takeaway home message, should I repeat or you already know? Experimental studies, observational studies, right? And based on what we need to select. Okay. So I hope this session makes sense to you. Let me tell you guys, whenever you read a research paper, do not immediately focus on the results only. Just remember that what study design was used and why, right? Was it appropriate for the research question? Like, what are the strengths and the limitation of this particular study? And can the findings be applied to my patients or not? So, if you can answer these questions, you have already developed one of the most important skills in the clinical research and evidence based pharmacy practice, right? So, this question should come into your mind definitely, right? So I hope the session makes sense to you. Any questions from your side? Yes, I will be very happy if you have any questions. Looking forward in time, looking back in time is retrospective. Yeah. Case control is completely different and retrospective is completely different, right? Retrospective, I think already someone is explaining that, looking back in time, first checking the outcome and then checking the exposure of it. That is retrospective cohort. And the case control starts with disease and looking backward, like outcome first and the exposure one. And that is, case control is very important for the rare condition, rare diseases, while retrospective cohort is not for rare conditions, right? That is the only difference. Yeah, cross sectional. Cross sectionals means only a snapshot of the people. Okay, snapshot of the prevalence check करने के लिए, right? For an example, if you want to check the prevalence, how many percentage of the people in this particular group develops hypertension. So for that you need a snapshot, nothing to go for exposure, nothing to check later in the future. Okay, no, nothing to go in backward direction, nothing to do. That is what the cross sectional means, population snapshot. For signal detection in pharmacovigilance, we use observational studies. Yeah, it can be used, it can be used based on the what are the objectives, what are the outcomes, depends. Okay. If any questions are there, guys, feel free to drop messages in the group or you can text me personally as well. I am just a message away, okay? Don't worry. And tomorrow your lecture will be at 10. Sorry, 10 a.m. It is in early morning, okay? 10 a.m. Please wake up and join on time, okay? It is in the morning, so you will be free or Sunday will be free later. My Sunday will also be, free. I will be also free. I hope the session is making sense to you and you are getting the things. I have another schedule, so I'm not giving you much time, I know, but feel free to drop messages if you have any query. I will, I would love to discuss all the time. I'm just a message away, guys, okay? Thank you so much for joining. And tomorrow we'll have the session again. I will be waiting for you at 10 a.m. And we will discuss much things in detail tomorrow because I have a good time tomorrow, okay? Thank you so much, guys, joining today. Thank you.